Hello and good day to everyone. Welcome to our lecture on cell biology. I am your Sir Nicolay Lumawan Martin, a Faculty of Medical Technology, Medical Laboratory Science at the University of St. Louis. When we talk about cell, we know that the basic definition or understanding of cells is that they are considered as the fundamental units of life. And this definition has been a cornerstone in the field of biology and medicine. The journey of understanding cells actually started in the late 1600s when English scientist Robert Hooke developed or observed a plant cell using a crude microscope. This initial observation or discovery actually set the stage for further advancements in understanding cell. And in fact, in the 19th century, German scientists Matthias Leiden, Theodor Schwann, and later on expanded by Rudolf Virchow, postulated or proposed the cell theory, in which the cell theory actually laid the groundwork for modern biology in medicine because it establishes that all living organisms are composed of cells, and these cells are or arise only from pre-existing cells. Cells now have three major regions or major compartments, in which we have your nucleus, your cytoplasm, and the plasma membrane. The nucleus is considered as the control center of the cell, and it houses the genetic material or the DNA. At the same time, your nucleus coordinates activities such as growth, metabolism, and reproduction. You also have the cytoplasm, which is a semi-solid or semi-fluid matrix, in which the cytoplasm is the area where most cellular activities would occur because it contains now different organelles like mitochondria, ribosomes, and the endoplasmic reticulum. Lastly, we have the plasma membrane or the cell membrane. And your plasma membrane or cell membrane forms the cell's outer surface or outer covering, which will now separate the cell's internal environment from its external environment. It's also notable that your plasma membrane or your cell membrane is a selective barrier, which means that it could regulate the flow of materials inside and outside the cell or into and out of the cell. Now, your cell, in order for us to examine it, we use an instrument that would magnify the image and allow us for better or greater visualization, and that would be your microscope. There are two major types of microscope that is used in medical laboratory science, and that would be light microscopy and electron microscopy. The light microscope would allow us to visualize the general features of cells. It actually uses light to produce an image. However, while it can visualize the general features of cells, it can have limited magnification. In fact, we have the most commonly used type of light microscopy, which is your bright field microscopy. Your bright field microscope would work by passing light through a specimen, which then appears dark against a bright background. And this technique that is used in bright field microscopy is ideal for us to observe stained or naturally pigmented specimens. To address the limited magnification of light microscopy, we also have the electron microscopy, in which the electron microscopy is used now to study the fine structures of cells, and it uses a beam of electrons to produce an image. We have two common types of your electron microscopy, namely the transmission electron microscopy and the scanning electron microscopy. The TEM uses a beam of electrons that passes through a thin specimen to produce an image, and it would provide detailed internal morphology and structural information of cells and tissues. So it sees through the parts of the cell. Therefore, given its principle, it is ideal for observing the fine cellular details and organelles. On the other hand, your scanning electron microscopy, or your SEM, would scan a focused electron beam across the surface of a specimen without passing through it. Therefore, the data that is being produced, or the image that is being produced, would be a detailed surface morphology and topographical images. And therefore, your SEM is useful for examining surface structures and the three-dimensional views of specimens. Now, let's talk about the cell membrane and the cytoplasm. The cell membrane, or your plasma membrane, is considered as a lipid bilayered structure which is visible under transmission electron microscopy. It is actually composed of an amphipathic lipid bilayer containing embedded integral membrane proteins with peripheral proteins attached to its surfaces. Your cell membrane or plasma membrane is composed primarily of phospholipids or lipids, which is approximately 45 to 50%. We also have cholesterol, which is another major lipid that we can see in cell membrane or plasma membrane. And we also have protein molecules, which is also about 45 to 50%, and carbohydrates, which is around 4 to 8%, which is found only on the outer surface of the plasma membrane or the cell membrane. Now, the most common feature of your plasma membrane or your cell membrane would be its lipid bilayer. It means that if it's a lipid bilayer, it's composed mainly of phospholipids which have amphipathic properties. It possesses both hydrophilic heads and hydrophobic tails. Your hydrophobic tails, from the word itself, hydrophobic, it's considered as water-fearing. Therefore, the hydrophobic tails will face inward and it will create the non-aqueous environment that will act as a barrier. While the hydrophilic heads will face outward because your hydrophilic heads are water-loving. So it would interact with the aqueous environment inside and outside the cell. Another major lipid that we can see in our cell membrane or plasma membrane would be cholesterol. Your cholesterol now plays a critical role in maintaining the fluidity of the plasma membrane. In fact, the cholesterol would help prevent the fatty acid chains of the phospholipids from packing together. So again, your cholesterol fits between the phospholipid. Okay? And therefore, with this function of the cholesterol, it keeps the membrane flexible, especially in varying temperatures. At the same time, it has a function on membrane stability. Your cholesterol actually adds structural integrity to the plasma membrane. By interacting closely with the phospholipids, again, it would prevent the membrane from becoming too fluid or too rigid. Also, your cholesterol would contribute to the selective permeability of the membrane by making it less permeable to very small water-soluble molecules that might otherwise pass through more freely. At the same time, your cholesterol is involved in the formation of lipid rafts. Lipid rafts are specialized microdomains within the plasma membrane that organize cell signaling and molecules. So that is the different functions of your cholesterol. You also have membrane proteins. Your plasma membrane or your cell membrane contains two proteins. That would be your integral membrane proteins and your peripheral membrane proteins. Your integral membrane proteins are embedded within the lipid bilayer since it is integral. And these proteins, therefore, span across the membrane and are involved in various functions like transport and also signaling. In fact, your integral membranes have two interior faces, which is notably known as your E phase, or your extracellular phase, and you also have your protoplasmic phase or your P phase. The E phase now is the outer surface of the plasma membrane that would face the extracellular environment. On the other hand, your P phase or your protoplasmic phase is the inner surface of the plasma membrane that faces the cytoplasm. We also have the peripheral membrane proteins. Your peripheral membrane proteins are attached to the surface of the membrane. And these proteins would play roles in signaling, maintaining the cell's shape or structure, and other functions. And your membrane proteins actually have various functions. It serves as marker molecules, attachment molecules, transport molecules, receptor proteins, and as well as enzymes. Your membrane protein would serve as marker molecules rather, in which these proteins, which is often your glycoproteins, serve as identity markers. They play a crucial role in cell recognition and immune response. It will help the body distinguish between its own cells and foreign invaders. At the same time, the membrane protein could become attachment molecules or attachment proteins. And these types of proteins now helps your cells adhere to each other. From the word attachment, it would help the cells adhere to each other and to the extracellular molecule or the extracellular matrix. These types of proteins would be very essential in maintaining now the tissue structure and facilitate also cell movement and signaling. Notably, we have your cadherins and your integrins. Your cadherins are proteins that attach cells to another cells, while your integrins would attach cells to your extracellular molecules. Your membrane proteins can also serve as transport proteins. When we say transport proteins, these proteins are actually involved in moving substances across the cell membrane. They would ensure, therefore, that the essential molecules such as we have ions, nutrients, and waste products are efficiently transported into and out of the cell. In fact, there are three characteristics that is important for its function. Number one would be specificity. It means that each transport protein would only bind to a specific type or a certain type of molecule or ion. At the same time, competition. This would mean that the result of molecules with similar shape binding to the transport protein. And we also have saturation. It means that the rate of movement of molecules across membrane is limited by the number of available transport proteins. We also have three major classes of transport proteins. We have your channel proteins, and these proteins form pores in the membrane that would allow specific ions or molecules to pass through by diffusion. They are selective, and can be gated. And the opening or the closing in response to stimuli. Again, they could be selective and they can be gated, so there is an opening or closing in response to certain stimuli. You also have your carrier proteins, or we can also call this as your transporters. These proteins would bind to specific molecules on one side of the membrane. It will undergo conformational changes or structural change, and it will release the molecule on the other side. They are very crucial in facilitated diffusion and active transport. You also have your ATP-powered pumps. These proteins, from the word ATP-powered, would utilize the use of ATP or cellular energy to move ions or molecules against the concentration gradient. An example of your ATP-powered pumps would be your sodium-potassium pump, which will maintain the cellular ion balance. Here, we can see that these are the different membrane proteins, your channel proteins. It will transfer small water-soluble molecules. It usually contains pores as a domain that will serve as ion selectivity filter. You also have your carrier proteins, which transfer small water-soluble molecules as well. They are highly selective in nature. In fact, some notable examples of your carrier proteins would be your sodium-potassium pump, as mentioned earlier. What does your sodium-potassium pump mechanism does? Your sodium-potassium pump will actively transport sodium ions out of the cell and potassium ions into the cell. And again, this will be crucial for ion balance, and in fact, this is also used in maintaining cell volume. Aside from sodium-potassium pump, we also have your hydrogen pump. It will transport your hydrogen ions or your protons across the membrane. And this processes is very important in acid-base balance. We also have, for example, glucose carriers. Your glucose carriers, there will be it will facilitate now the transport of glucose into the cells using your GLUT proteins or GLUT transporters. And of course, we also have your ATP-powered pumps, in which it will require ATP molecules or the expenditure of energy for it to function. Next is we also have here your figure 3.5, in which it talks click and gated membrane channels. Your channel proteins are integral membrane proteins that create a small passage through the plasma membrane. The outside of these proteins is hydrophobic, which will match therefore the membrane's interior, which is hydrophobic. So the inside of the channel is hydrophilic naman. It will therefore allow ions or small molecules to pass through it. Only ions or molecules with the right size, charge, and shape can actually move through the channel. And the specific charges inside the channel would determine which ion can pass. Okay? And if we try to connect this to a pathophysiologic case, your different channels, ion channels, the functioning of your membrane ion channels is actually vital to equilibrium or homeostasis that can occur if a type or actually if there is an imbalance. It could actually lead to a dysfunction, or it could actually lead to a serious disease. And one of the serious disease could be your cystic fibrosis. Your cystic fibrosis is a genetic disorder that affects specifically your chloride ion channels. The failure of these ion channels to function will cause the affected cells to produce thick and viscous secretions. Again, thick and viscous, malapot na secretions. Although in your cystic fibrosis, it affects many types of cell, its most profound effects are in organs such as your pancreas and lungs. So what happens? In your pancreas, the thick secretions would block the release of digestive enzymes. And because there is a blockage in the release of your digestive enzymes, it would result now to inability to digest certain types of food, and sometimes could actually lead to serious cases of pancreatitis. And again, When we talk about pancreatitis, itis, or titis, that would be the inflammation of your pancreas. Aside from your pancreas, we also have mentioned that it could actually have profound effect in your lungs. So what happened? Or what happens rather? In the lungs, the stick secretion could actually block the airways. And of course, individuals who are diagnosed therefore of cystic fibrosis would have a difficulty of breathing because the stick secretions again would block the airways. Okay? Now, we also have your membrane proteins as receptor protein. Receptor proteins now are involved in cell signaling. They bind to specific molecules such as we have your ligands, or we have hormones or neurotransmitter, and it will trigger now a response within the cell. Okay? So when a ligand binds to these receptors, it directly or it could also indirectly cause the associated channel to open or close, regulating ion flow into or out of the cell. And upon ligand binding, this receptor will activate your G proteins, which can now trigger a cascade of intracellular signaling events that would lead to cellular responses. Now we have here in figure 3.8 the opening and closing of a ligand channel protein. When a specific molecule or your ligand, for example, would bind to a ligand channel protein on the cell membrane, it will now trigger a change in the protein shape. This change can either open or close the channel within the protein. Therefore, it will control the flow of ions. And definitely, it will control the flow of ions into and out of the cell. Now, the opening of the channel will therefore allow ions to pass through, while on the other hand, closing or the closing of the channel would definitely prevent the movement of the molecules inside or outside the cell. Thereby, it will regulate the various cellular processes. For example, nerve signaling, or also we have your muscle contraction. In figure 3.9, we have here receptor link to a G protein complex. So when a signaling molecule such as your ligand binds to a receptor that is linked to a G protein complex in the cell or on the cell membrane rather, it will activate the receptor. And through the activation of the receptor, it will cause again the change of shape. And this activation would trigger the G protein inside the cell to bind to GTP. Your GTP now is a type of energy molecule. And if it will bind to your GTP, it becomes activated. This activated G protein now moves along the inside of the membrane to influence other proteins or enzymes. It will therefore initiate a cascade of cellular responses. And this process amplifies the signal from the original ligand, which would lead to various effects inside the cell. Lastly, we also have your membranes as enzymes. When we say enzymes, enzymes actually play a lot of roles or various roles in the body. Your enzymes now, specifically your membrane-associated enzymes, will catalyze chemical reactions at cell surface. They can be involved in different processes. For example, signal transduction. We also have metabolism, Also, it could be involved in the breaking down of specific substrates. Again, when we talk about your enzymes or your membrane-associated enzymes, it will catalyze chemical reactions either inside or outside the cell. Now, let's talk about the movement of through the cell membrane. The cell membrane separates now your extracellular material, or when we talk about extracellular material that's outside the cell, from the intracellular material or inside the cell, and is selectively permeable. It means that it controls what enters and exits the cell. Okay? So inside the cell, there are higher concentrations of enzymes, other proteins, glycogen, and potassium ions. In the extracellular, outside the cell, there are also higher concentrations of sodium, calcium, and chloride ions. Okay? We also have different types of molecule movement. If we're talking about lipid-soluble molecules, like substances like your oxygen, carbon dioxide, and steroids, they can easily pass through the plasma membrane by dissolving in the lipid layer. If we're talking about small non-lipid molecules, these molecules like your urea can diffuse between the phospholipid molecules of the membrane, but the movement could be limited. Okay? And if we're talking about large non-lipid soluble molecules and also ions, this cannot pass directly through the lipid bilayer. And therefore, if it cannot pass directly, it would require the help of transport proteins to move across the membrane. Okay? And large particles and whole cells can be also engulfed and transported across the membrane in the form of vesicles. Your vesicles now are considered as small membrane-bound sacs. Okay? And again, if we're talking about selective permeability, this is a very important property that is crucial for maintaining the internal environment of the cell. Okay? So it will allow essential nutrients in while it will keep also harmful substances out of the cell. At the same time, if we're talking about transport proteins, this include your channel proteins, your carrier proteins. They will assist now in the moving of substances that cannot diffuse directly through the lipid bilayer. And again, through your vesicle transport, these are processes like endocytosis or exocytosis, which involves the use of vesicles to move large molecules. Okay? So it moves your large molecules such as, for example, your nutrients or your waste products across the membrane, which will maintain cellular function and communication. The first one is the different processes. Okay? So we have what we call as passive processes and active processes. We first discuss passive processes. Your passive processes is a movement of substances down their concentration gradient without the expenditure or the utilization of cellular energy or the ATP. We have what we call now as diffusion. Your diffusion is defined as the net movement of particles from an area of higher concentration to an area of lower concentration. We actually have two common types of diffusion. We have your simple diffusion and your facilitated diffusion. Your simple diffusion is an unassisted movement of small non-polar substances across a selectively permeable membrane. Example are the oxygen and your carbon dioxide. Okay? Again, simple diffusion, unassisted movement. When we talk about facilitated diffusion, we're going to talk about your movement of ions and molecules that are polar in nature down the concentration gradient using now transport proteins. And in diffusion, we have the following terminologies. First is we have solute. Your solute now, that's the substance that is being dissolved. You also have solvent, the dissolving medium. And when we talk about concentration gradient, that's the concentration difference when solutes are not evenly distributed in a solvent. Now, if we try to look on the factors that influence diffusion, these are the following factors. Number one, the concentration gradient magnitude. So as rate of diffusion increases, always remember that the diffusion gradient also increases. Therefore, the rate of diffusion and diffusion gradient is directly proportional. Its relationship is directly proportional. We also have the temperature of the solution size of diffusing molecules. We also have viscosity of the solvent. When we talk about viscosity, that's the measure of the fluid's resistance to flow. Or, kumbaga in Filipino, kumbaga it talks about kung gaano kalapot ang isang fluid. And therefore, if a fluid is highly viscous, there is less flow, okay? On the other hand, if a fluid is less viscous or non-viscous, therefore, the fluid will flow more. And the diffusion occurs more slowly, therefore, in viscous solvents than in watery solvents. Okay? Now, we have here the diffusion through the plasma membrane. As we have mentioned, your diffusion now is an important means by which your substances would move between the extracellular and intracellular fluids. Okay? We also have the concept of osmosis. Your osmosis, on the other hand, is actually a special case of facilitated diffusion. It involves now the movement of water molecules specifically through aquaporins from an area of lower solute concentration to an area of higher solute concentration. We also have definition of terms such as we have your osmotic pressure. When we talk about osmotic pressure, this is already the force needed to stop water from moving across a selectively permeable membrane by osmosis. The higher the osmotic pressure of a solution, the greater its tendency to draw water across the membrane. In fact, we have these following terms. Isosmotic or isosmotic, we have hypersmotic, and we have hyposmotic. Your isosmotic, this is when two solutions have the same concentration of solute particles. Therefore, if they have the same concentration of solute particles, they have the same osmotic pressure, meaning the water does not move between them by osmosis. We also have hyperosmotic. When we talk about hyperosmotic, a solution with higher concentration of solute particles would have higher osmotic pressure compared to the another solution. This means that it will draw water towards itself if separated by a selective permeable membrane. And lastly, we have your hypoosmotic, in which in hyposmotic, a solution is more dilute. Okay, so it's a more dilute solution with fewer solute particles, and therefore it has lower osmotic pressure as compared to a more concentrated solution. So it will lose water to the more concentrated solution if they are separated by a selectively permeable membrane. Okay, and when we talk about tonicity, tonic, it's a condition where a cell's shape remain constant, maintaining its internal tension or tone. If it's osmotic, again, it refers to the concentration of a solution. We have here in figure 3.13 the different scenarios of the effects of hypotonic. We have your isotonic, and we also have your hypertonic solutions. Definitely, if it's an isotonic solution, again, it's equal, it's constant, neither of your blood cell or specifically your erythrocytes would shrink or swell. If it's hypertonic, your water will move out from cells by osmosis. Therefore, the red blood cells or the erythrocytes would actually shrink, or there is granation. And if it's hypotonic, the water now will move into the cell by osmosis. Therefore, there is swelling, or even in worst case scenario, it will burst or it will lyse, or if there is lysis in a hypotonic solution. We also have your active processes. Of course, when we talk about your active processes, this is already the movement of substances against their concentration gradient that would require already your expenditure of cellular energy, or usually we have your ATP. And when we talk about your ATP, we have your ATP-powered pumps. So these pumps will transport ions or molecules actively across the membrane. Example would be your sodium-potassium pump. And again, we again have your vesicular transport as well, in which this is the movement of substances into or out of the cell via vesicles. We also have your secondary active transport. It involves inactive transport of an ion such as sodium out of a cell, which will establish a concentration gradient with a higher concentration of ions outside the cell. Okay? And we also have your vesicular transport, in which we already mentioned, and your endocytosis. In your endocytosis is actually a process of internalizing substances from extracellular environment. We have three types of endocytosis, namely your phagocytosis, pinocytosis, and your receptor-mediated endocytosis. For your phagocytosis is considered as clathrin-independent uptake of large particles or cells. For example, we have here in the immune cell engulfs a pathogen. Okay? You also have your pinocytosis. Your pinocytosis is also a clathrin-independent uptake of extracellular fluid and dissolved fluids. And we have your receptor-mediated endocytosis. This is considered as clathrin-dependent uptake of your specific molecules bound to the cell surface receptors. For example, the uptake of your cholesterol via the low-density lipoprotein receptors. Okay? So that would be your endocytosis. We also have the concept of your exocytosis, in which in exocytosis, that's the process already of expelling the substances from the cell via vesicles, in which we have two pathways. We have the constitutive pathway and we have your regulated pathway. It's a simple that if we talk about your constitutive pathway, that's a continuous non-regulated secretion of materials. And if it's regulated pathway, definitely the secretion of the substances is in response to specific signals. Okay? Next is we also have your cytoplasm. In your cytoplasm, I would like to highlight the semi-fluid structure, or the semi-fluid matrix. And we call this as your cytoplasmic matrix. Your cytoplasmic matrix will house the various organelles and inclusions. Your cytoplasmic matrix contains variety of solutes such as your sodium, potassium, calcium, and you also have organic molecules such as your intermediate metabolites, your carbohydrates. We also have lipids, we have proteins, and your RNA. And within your cytoplasm, your organelles are either characterized as membranous or non-membranous. If we're talking about membranous, these are surrounded by membranes, and it would include structures such as your mitochondria, your Golgi apparatus, and you also have your lysosomes. For non-membranous, therefore, this lacks a membrane. So it would include your ribosomes and also the cytoskeleton components, for example. Okay? And speaking of cytoskeleton components, your cytoskeleton would support now the cell by holding the nucleus and other organelles in place. So it plays a key role in cell shape changes and the movement of organelles within the cell. In fact, your cytoskeleton is composed of three groups of proteins. Namely, we have your microtubules, your actin filaments, and your intermediate filaments. Your macrotubules now provide structural support, and it will serve as tracks for the movement of organelles. Your actin filaments, on the other hand, is involved in cell movement and shape changes. And lastly, your intermediate filaments would provide mechanical strength and would also help in the maintenance of cell integrity. Again, we also have your cytosol. We forgot the cytosol, in which your cytosol, again, is your fluid portion of the cytoplasm. So it's usually colloid and viscous in nature. Okay? Next is we have your group of proteins. Again, as we have mentioned, your microtubules. What are the different functions? It will serve as intracellular vesicular transport. It also helps in the movement of your cilia and flagella. Also for the attachment of chromosomes to the mitotic spindle. It also serves in the elongation of cells and movement, and also the maintenance of your cell shape. Your actin filaments, or your microfilaments rather, it has a function on the anchorage and the movement of your membrane proteins, the formation as well of your structural core of microvilli. It also serves in the locomotion of cells, and also it serves as extension of cellular processes. And lastly, your intermediate filaments, it's like a rope-like filaments in which it has an intermediate size now between your microtubules and microfilaments. What are the usual functions of your intermediate filaments? They are for mechanical support or mechanical strength to cells. Also, we have for cell junctions. Okay. Next up is we have your cytoskeleton inclusions. Your cytoskeleton inclusions, this is contained in the cytosol that are aggregates of chemicals either produced or taken in by the cell. Examples would have your lipid droplets or your glycogen granules, hemoglobin, your melanin, and your lipochromes, for example. And we also have your nucleus and your organelles. Again, if we're talking about your organelles, these are the different structures now that we can see within the cells that have specialized functions. Okay? And your nucleus, okay, focusing on nucleus, your nucleus now is a large membrane-bound structure that is typically found in your center of the cell. It actually have various shapes. It could be spherical, elongated, or lobe, depending now on the cell type. And for most of the cell's life cycle, chromosomes are spread out within the nucleus as thin threads, which is known as your chromatin. Okay? Next is we have your deoxyribonucleic acid, or the DNA, which is mostly found, again, within the nucleus. Okay? Your DNA and associated proteins, such as your histones, are organized into discrete structures called chromosomes. Your DNA would determine the structural and functional characteristics of the cell. It's also a large molecule that does not leave the nucleus but functions by means of its intermediate, which is your RNA, which can actually leave the cell through the nuclear pores. And it also determines the structure of your mRNA or your messenger RNA, the ribosomal RNA or rRNA rather, and your the RNA or the transfer RNA. And your gene now is defined as a region of DNA molecule that specifies an RNA molecule. I'd like to highlight on histones. In biology, in fact, your histones are highly alkaline proteins that are found in eukaryotic cell nuclei. They your histones package and order the DNA into structural units which we call as your nucleosomes. Okay? Your histones actually have 5 major families. Namely, we have your H1 or H5, your H2A, H2B, you also have your H3 and H4. Your histones, H2A, H2B, H3, and H4, as known as your core histones. When we're talking about core histones, what are core histones? These are central proteins around your DNA, okay? Or they are central proteins around where DNA or which DNA binds to form the nucleosome core. On the other hand, your H1 or H5 are your linker proteins, okay? Or your linker histones rather. Your linker histones is a protein that would bind to the DNA which between your nucleosome, again, it will bind to the DNA between your nucleosome and it will help in the stabilization of the higher order structure of chromatin. Okay? And during cell division, the thin chromatin filaments condense and it will coil tightly to form your compact structures which is known as your chromosomes. And this dense coiling ensures that your genetic information or your genetic material rather is efficiently organized and there is an accuracy in the distribution of the daughter cells. Okay? Next is we also have your cells without nuclei. The cells lacking a nuclei can still carry out protein synthesis using mRNA that was produced before the nucleus degenerated. And protein synthesis continues as long as this mRNA can be translated by ribosomes. Okay? You also have your organelle nucleolus. Your nucleolus now is a dense region. Okay, so this is not, rather, to correct it, this is not an organelle, rather, it is a region, it's a dense region within the nucleus that lacks a surrounding membrane. Your nucleolus is responsible, okay, in manufacturing ribosomes. And your nucleolus contains portion of chromosomes with DNA that is transcribed into ribosomal RNA, which is again essential for ribosome assembly. You also have your ribosomes. Your ribosomes are either free or attached to the endoplasmic reticulum. Your ribosomes, again, its major function is for protein synthesis. For free ribosomes, these are scattered throughout the cytoplasm and they are synthesized or they synthesize proteins that are used within the cell. Again, they use or they synthesize proteins within the cell. On the other hand, the ribosomes studied or attached to the endoplasmic reticulum are usually in the RAF-ER specifically, they would produce integral membrane proteins and proteins that are secreted from the cell. Okay? Now, since we have talked about your endoplasmic reticulum, your ER is a convoluted membranous network that actually extends from the surface of the nucleus to the cell membrane. It encloses a series of interconnecting or intercommunicating channels and sacs and tubules which we call as your cisternae. And your cisternae is in fact the interior spaces of the sacs and tubules. What are the functions of your endoplasmic reticulum? Your endoplasmic reticulum plays a crucial role in the synthesis of your proteins and lipids. At the same time, it's also important in transport because your ER would serve as a pathway for the transport of materials within the cell from the nuclear envelope to the cell membrane. Okay? And as we have mentioned, your ER have two types in which you have your RER or the rough ER, which consists of sac-like and parallel stacks of latent cisternae. Again, it is studded with attached polyribosomes. And this attachment of your polyribosomes would give it its rough appearance. What are the functions of your RER? It's for the synthesis of proteins that are not destined for the cytosol, including those meant for secretion or incorporation into the cellular membranes. Okay? It also begins the process of glycosylation for glycoproteins. And it also performs modifications on newly formed polypeptides. So it includes folding and assembly into multi-chain proteins. Okay? So again, those are the functions in protein synthesis and segregation, in initial glycosylation, and in the post-translational modifications. Your soft endoplasmic reticulum, on the other hand, have the following functions. The SER would manufacture lipids, which includes your phospholipids, your cholesterol, your steroid hormones, as well as your carbohydrates. And the enzymes required for these processes are embedded in your soft endoplasmic reticulum membrane. So it's used for lipid and carbohydrate synthesis. Aside from that, your SER is involved in detoxifying harmful substances such as drugs and metabolic waste products. So it's involved, or it participates in detoxification. And lastly, it in your skeletal muscles, okay, in skeletal muscle cells, your SER rather, could store calcium ions. So these are crucial for muscle contraction. So those are the notable functions of your SER. It's for lipid and carbohydrate synthesis. It participates in detoxification and calcium storage, specifically in your skeletal muscle cells. You also have your Golgi apparatus or your Golgi body. Your Golgi apparatus is actually named after Camillo Golgi, who discovered it in 1898. Your Golgi apparatus is composed of smooth membranous sacules containing enzymes. What are the functions of your Golgi apparatus? Your Golgi apparatus further modifies proteins after their synthesis in the endoplasmic reticulum. Therefore, your Golgi apparatus has the function of completing the post-translational modifications. Aside from that, of course, the classical function would be it is used for sorting and packaging proteins for transport to their correct or designated destinations later on within or outside the cell. So it will package and addresses proteins. So those are the functions of your Golgi apparatus. We also have your secretory vesicles. Okay, these are, or their membrane, fuse with the plasma membrane, and the contents of the vesicles are released to the exterior by exocytosis, in which you have discussed a while back. And the membrane of the vesicles are then incorporated into the plasma membrane. You also have your lysosomes. Your lysosomes are actually spherical. They are around 0.05 to 0.5 micrometer in diameter. They are present uniformly, okay, or they are like a uniformly granular. They have a uniform granular appearance rather in your TEM or transmission electron microscopy, and your lysosomes are membrane-limited vesicles that contains approximately around 40 Okay, again, around 40 hydrolytic enzymes, which is responsible for degrading various biomolecules. Okay, so in lysosomes, I would like to insert a pathological case in which some diseases result from a non-functional lysosomal enzymes. Kasi again, if we talk about your lysosomes, there is around your lysosomes are vesicles that could contain at least or approximately 40 hydrolytic enzymes. So there are some, again, diseases that result from non-functional lysosomal enzymes or deficiency of such enzymes. For example, your Tay-Sachs disease. Your Tay-Sachs disease is a rare genetic disorder in which there is an inability of lysosomal enzymes to break down your gangliosides. Okay? Your gangliosides now is a specialized membrane lipids of your neurons. And these lipids would therefore accumulate in the cells and will cause damage. The common symptoms of your Tay-Sachs disease would include paralysis. We also have blindness. And in worst case scenario, death, usually before the age of 5. Okay? Again, before the age of 5. And damage, therefore, to the CNS would begin in fetus. But other symptoms do not become apparent until several months after birth. So the infant will appear to develop normally, but as time goes on, it begins to regress. It will lose its motor function, it would lose its vision, it will experience seizures, and it will actually exhibit delayed mental development. So there would be mental retardation in the case of a patient with this disease. And as a result, the destruction of the cells in the nervous system, definitely it could lead to death. So the individual or the patient with Tay-Sachs disease or diagnosed with this type of disease usually dies at the age of 4 or 5 years old. And unfortunately, up to now, there is no known cure or treatment for Tay-Sachs disease. However, we have genetic counseling of high-risk couples, which is accompanied which are accompanied with prenatal screening for those that are greatly reduced or to help in reduction of the incidence of the disease. Okay, so that's a pathophysiological case of your lysosomes. We also have your peroxisomes. Your peroxisomes now are spherical organelles enclosed by a single membrane. It's similar to the structure of lysosomes, or lysosomes rather, but they do not contain hydrolases or lysosomal hydrolases. What are the functions? Of course, oxidation reactions. Okay, if we say oxidation reactions, we have enzymes such as your oxidases. Your oxidases now are enzymes that will remove your hydrogen atoms from various substances and will transfer them to the molecular oxygen. And therefore, it will produce your H2O2. Your H2O2 is your hydrogen peroxide. At the same time, we also have your catalase. Your catalase now is an enzyme that would break down your H2O2 or your hydrogen peroxide into water and oxygen, thereby preventing the damage to the cell. What is the significance of your peroxisome? It's significant as it plays a crucial role in metabolism, specifically in cellular metabolism, because it participates in oxidative reactions and it will detoxify harmful substances. We also have your proteasomes. Your proteasomes are large protein complexes that contains enzymes responsible for breaking down and recycling proteins within the cell. So they degrade proteins, tagged for destruction by ubiquitin. Okay, again, they will degrade protein that is tagged by ubiquitin. And this is a process now that is crucial for regulating various cellular processes and maintaining protein quality. And of course, we have your mitochondria. Your mitochondria, of course, is the powerhouse of the cell, but I'd like to focus more of the theory or the hypothesis that goes around mitochondria. your mitochondria actually is related to your endosymbiont theory. Your endosymbiont theory would propose that certain organelles, for example, in your eukaryotic cell, we have your mitochondria rather, and also your chloroplast. They are originated from free-living prokaryotic organisms that were engulfed by primitive eukaryotic cells. So it's believed that your mitochondria are or have originated from proteobacteria. Okay? So your mitochondria are believed to have originated from proteobacteria. And the engulfed prokaryotes will form a symbiotic relationship with the host cell, which provides advantages such as enhanced ATP production for your mitochondria, and in the case of your chloroplast, it's photosynthesis. What are the evidences that this theory could actually be true? We have the double membranes. We have to know that your mitochondria actually have a double membrane. So it's consistent with the engulfment. Aside from that, the genetic material. These organelles, your mitochondria and your chloroplast, contain their own circular DNA, which is quite similar to your bacterial DNA. And they also have their own ribosomes, which is also similar again with the bacterial ribosomes than to your eukaryotic ribosomes. At the same time, your mitochondria could produce independently of the cell through a process which we call as your binary fission, which is already akin to your bacterial replication. Therefore, your endosymbiont theory would explain the origin of eukaryotic cells and the presence of organelles with prokaryotic characteristics within them. And that's the case of your mitochondria and your chloroplast. We also have your centrosome. Okay? Your centrosome are specialized area of cytoplasm near the nucleus that would serve as the center for microtubule formation in the cell. It's actually crucial for determining the cell shape and movement. You also have your centrioles, which is located within the centrosome, and there are typically two centrioles oriented perpendicular to each other. And they play a key role now in organizing microtubules and are involved as well in cell division. Okay? We also have your cilia. Your cilia are hair-like processes. They are very numerous, in fact, in your epithelial cells of your upper respiratory tract. Cilia can also be seen in the parts of the female reproductive tracts, even in the ependymal or ependymal lining of the cavities of the central nervous system. We have here gynine arms and your basal body. Your gynine arms are proteins that connects the adjacent pairs of microtubules and push them past each other. This movement is essential for beating of the cilia and flagella. We also have your basal body or the modified centriole. The modified centriole located in the cytoplasm at the base of the cilium or flagellum. And your basal body acts as an anchoring structure. It will organize the microtubules that extend into the cilium or flagellum. And you also have your flagella, of course. Your flagella are long, semi-rigid, helical, hollow tubular structures, mostly composed of proteins, which is your flagellin, okay? And they show undulating or wave-like motion, okay? Or wave-like movement. You also have your microvilli. Your microvilli, these are cylindrically shaped extensions of the plasma membrane of typically about 0.5 to 1 micrometer in length and 90 nanometers in diameter. So they do not move, okay? And they are supported by actin filaments rather than microtubules. And your microvilli would therefore increase the surface area of the cell membrane. It will enhance its ability to absorb nutrients, and it could also interact with the environment. Okay? So again, it will enhance the ability to absorb nutrients and interact with the environment. Now we have your whole cell gene activity, or your whole cell activity rather, in which we have your gene expression. Your gene is considered or is termed as the functional unit of heredity that carries now, of course, the genetic information for traits and characteristics. When we're talking about heredity, this is the process in which your genetic traits are passed from parents to their offspring. We also have gene expression. If we're talking about gene expression, that's already the process through which the information stored in DNA is used to produce RNA and or proteins. And if we talk about the genetic code, that's the information encoded in messenger RNA or mRNA that links the nucleotide sequence of mRNA to the amino acid sequence of a protein. In fact, there are gene expression. In gene expression, rather, it involves two steps or two key steps, and that would be transcription and translation. In transcription, this is already the process of copying a segment of DNA into RNA, and in translation, the process wherein your RNA sequence from transcription is used to assemble amino acids into a protein. Okay? And if we're talking about life cycle, we have two stages, and that would be your interphase and your cell division. Okay? In which in cell division, it would include your mitosis and your cytokinesis. Okay? In your interphase, okay, let's try to discuss. Your interphase, this is already a phase between your cell divisions, where a typical cell spends most of its life cycle. During this phase, the cell would prepare to divide by increasing in size. Okay, so it will double its components or its organelles, and it will also replicate its DNA. The centrioles within the centrosomes are also duplicated. So those are the things that happens in your interphase. Again, it increases in size, it doubles its components or your organelles and your inclusions. It will replicate the DNA, and as well as the centrioles within the centrosome are also duplicated. There are subphases of your interphase. First is the first gap or gap 1 phase, in which the routine metabolic activities and cellular functions happens in this G1 phase. And the duration is approximately 11 hours. We also have your S phase or your synthesis phase, in which the DNA replication occurs here in this phase, where again new DNA is synthesized, and it takes about 8 hours. And you also have your gap 2. Your gap 2 now, it is the gap phase in which the cell will prepare for cell division, in which it will prepare for cell division, and it ensures now that all components, all the organelles and inclusions inside the cell are ready. And the duration is about 4 hours. Okay? And also, please take note that we also have your gap 0. Your gap 0 is where your cells are resting. So they do not divide unless they are stimulated. At the same time, please take note that in the interphase, during interphase rather, okay, there is a secretion of enzymes. So it occurs during interphase as part now of routine cellular activities. Okay? And then we have your DNA replication. Okay? That's the process already in which two strands of DNA serve as template for making complementary new strands of nucleotides, in which we have the following. Your DNA polymerase, this is now a type of enzyme that would add new nucleotides to the 3' end of the growing DNA strands during the replication. The leading strand, that's the DNA strand that is synthesized continuously in the direction of the replication fork. The lagging strand, that's the DNA strand that is actually synthesized in the short segments known as your Okazaki fragments, which are later joined together. And we also have your DNA ligase. Your DNA ligase is now an enzyme that will splice or will join the Okazaki fragments on the lagging strand to create a continuous DNA strand. Okay? And if we try to differentiate your DNA and RNA, these are the different points of differentiation. Number one is your structure. Definitely your DNA or your deoxyribonucleic acid has a double-stranded helix structure. It has a sugar phosphate backbone. Okay? Again, it has a sugar phosphate backbone. And it also contains nitrogenous bases. Okay? Your RNA, it's a single strand, okay, molecule with a sugar phosphate backbone and nitrogenous bases as well. So that would be the structure. So again, double helix for DNA, single-stranded for your RNA. For the sugar component of both your DNA and RNA, the sugar component of DNA, it contains your deoxyribose sugar. On the other hand, your RNA contains your ribose sugar. In the nitrogenous bases, your DNA includes your thymine, while in your RNA, it includes your uracil instead of your thymine. What are the functions or the difference in function? Your DNA stores and transmit now your genetic information. Therefore, your DNA will serve as the blueprint for all cellular functions. On the other hand, your RNA can act as messengers, okay? It will carry the genetic information from DNA to the ribosomes for protein synthesis and has other roles such as in your tRNA and rRNA, which is also involved in protein assembly. And if we talk about its stability, your DNA is more stable and durable. Okay? Therefore, it's suited for long-term storage of genetic information. Unlike for your RNA, in which it's actually less stable and it's more prone to degradation. So it's suited only for short-term tasks, like for example, as we have mentioned for protein synthesis. Okay? Now, in cell division, there are two major events. It would be your mitosis and your cytokinesis. In your mitosis, that's already the process in which your chromosomes are divided and distributed into two nuclei. Okay? And this process will ensure that each new nucleus will receive identical sets of chromosomes. On the other hand, your cytokinesis, that's already the division of your cytoplasm that follows actually after mitosis. So it results in the separation or the formation of two separate cells in which each contain one of the newly formed nuclei. Okay? In your mitosis, we have different phases. Again, we have already discussed the interphase in which we have your gap1, S phase, and gap2, and your resting phase, which is your gap0. You also have your M phase, okay? So your mitosis phase, in which this is already the stage of cell cycle in which the nucleus will divide into new cell, or rather, it will divide into new nuclei. Okay? So each with, again, the same amount and type of DNA as to its parent nucleus or original nucleus. Okay? So this phase actually takes approximately about an hour, and it includes the following components. We have your mitotic chromosomes, your chromatid, and your kinetochore. Your mitotic chromosomes are highly condensed chromosomes which are visible, okay, during mitosis. And each consisting of two identical sister chromatids. Your chromatids, these are one of the two identical halves of the duplicated chromosome, which is joined together at the centromere later on. And your kinetochore now is a protein structure that, or on the centromere of each chromatid, where the spindle fiber attach during the mitosis to pull the chromatids apart. Okay? Next is you also have your, okay, after the M phase, in your mitosis, diba? We have your prophase, we have your metaphase, your anaphase, and your telophase. In your prophase, just a summary or just a recap or a review from your basic education biology. In your prophase, your chromosome condense, the spindle fiber will form, and the nuclear envelope will break. In your metaphase, your chromosome will line up at the equator or the equatorial plane. In anaphase, the sister chromatids are pulled apart toward the opposite poles of the cell. And in your telophase, the chromatids now reaches the pole after pulling apart, and the nuclear envelope forms around each set of chromosomes. Okay? So that's PMAT. You have prophase, metaphase, anaphase, and your telophase. You also have your cytokinesis. And again, as we have mentioned, your cytokinesis is the division of your cytoplasm, which results in two separate cells in which each one would have one nucleus. And the first sign of your cytokinesis would be the formation of the cleavage furrow. So that would be for cytokinesis. We also have the concept of your apoptosis. Your apoptosis is your programmed cell death, in which it's a controlled process by which the number of cells in tissues is regulated and adjusted. What would be the process of apoptosis? Your apoptosis, okay, during apoptosis, the cell would undergo a series of steps leading to their self-destruction in a regulated manner. So from normal cell to shrinkage, there is blebbing, okay? The cells will break into apoptotic bodies, and it could lead to phagocytosis, okay? So the cells will break down into smaller fragments or into apoptotic bodies. And the last step would be phagocytosis, in which in the phagocytosis, the specialized cells such as your macrophages will engulf and digest these cell fragments or these apoptotic bodies, which will prevent inflammation and will maintain tissue health. Okay. Next is we have cellular aspects of aging. These are the different major hypotheses of aging. First is the cellular clock. In the cellular clock, this is a theory which is proposed that cells have limited number of divisions before they stop dividing due to telomere shortening. So it leads to cellular aging. Again, there are limited number of divisions before they would stop dividing due to the telomere shortening. We also have death genes. Your death genes would suggest that there are certain genes that are programmed to trigger aging and death processes in cells that would contribute in the aging of the organism. You also have DNA damage. This indicates that the accumulated damage to DNA over time from environmental factors, or there are cellular processes that could damage the DNA, would lead to aging and cellular dysfunction. We also have your free radicals. It's been proposed that damage caused by your free radicals, what are the free radicals? These are considered as unstable molecules that could actually cause severe damage to the cellular components. This will contribute as well into the aging and degenerative diseases. And lastly, we have your mitochondrial damage, in which in your mitochondrial damage, this suggests that the damage to mitochondria, which is the energy house or the energy producing structure in the cell, would affect the cellular function and contribute to the aging process. So those are the different major hypotheses of cellular aging. Okay, so that's it for your cell biology, in which we have been able to study the different structure, the different functions, and as well as the cell cycle and cellular aging, or the different theories of cellular aging.